Okay, so thank you everyone. This is my favorite photo from it's quite some time ago actually, but this is kind of as Sharon said, I am from Hilo, and so this is kind of the environment I grew up in, where we would go to the at that time HAIA shows and everything, and so when I went to the mainland, of course, this is the photo I took with me just to remind me that this is Hawaii and we're from in Hilo. So. Okay, so today what we're going to give you is an update on a USDA specialty crop block grant that was given through Sharon's department, actually, at the Hawaii Department of Ag. And so what we're doing, and what Tessie had mentioned earlier, is that we're using, she had spoken about conserving germplasm of dendrobiums, but this is conserving germplasm of anthuriums. So you have both programs going. And so part of our unit is the germplasm repository. So we kind of know the importance of germplasm with the events in Puerto Rico and Miami, which is which are our counterparts in the USDA ARS system. We know how important it is also to have a backup as well. And so catastrophic events can happen at any time, and so you want to not put all your eggs in one basket, basically. And so basically, as Tessie had also mentioned, is that fashion, colors and everything is kind of fashionable. So it would be nice to have it in tissue culture already, so that you can move faster to try and propagate it a little bit faster than rather than trying to initiate it every time. Okay. So these are collaborators, and so Tessie as well as Dr. John Suzuki. And so I'll kind of go over some of the collections. So Tessie had mentioned how difficult it is to collect new species now. And so, and I kind of wanted to highlight this because John has worked really, really hard on getting some of these last species around, because he has to sign all types of agreements, because sometimes they're only in a botanical garden, and because you have different clauses where everyone has protective rights against all these species. He has to work very, very hard to get some of these. And so what we're going to do is try and have these in the greenhouse, as well as backing them up in tissue culture, and that way we can kind of protect them from diseases. Yeah, so some of these are replicated both here and as well as with Tessie and Manoa. So just going over some of the species is Amnicola, which everyone knows as a breeding population. Actually, Beckerii. I'm sorry if I pronounced the Latin names a little wrong. Anthurium Clidemodis pacificum. And so you can see kind of the diversity in the shapes and the forms. A little bit more, almost like a pubescence on this type. Chamberlainium, and so you can see the flowers, both the mature as well as forming seed pods. Okay. Coriaceum, and so you can see the different types. More of an upright type of foliage. Corallium. Okay, for Anthurium formorsum. I thought I had a thing. And Fragrantissimum. So these are kind of like your potentially untapped resources for future breeding, for future different novel traits. Hoffmanii. Okay. Lindanianum, which has a kind of nice growth. Nymphaliifolium and Pedigeranum, Anthurium radicans. Okay. Anthurium rectavum. Anthurium reflexinarium. I should have taken Latin as my second language in high school, I think. Anthurium scandens usually has a nice purple berries on the fruit, on the spadix. What's that? Yes, it's a lot more viney type of growth, or long, elongated growth. Anthurium beechii. Okay, Anthurium Warockianum. There are certain ones that it's like, oh my gosh, Anthurium wendeligeria. So some of these are the last ones that they have at the botanical gardens. And so either to get specimens, or John had to, sometimes they would only give him DNA and things. So these are kind of a valuable resource that we're trying to preserve. Okay, and then at the same time, what we're also trying to do is preserve some of the heirloom varieties, so varieties that might have departed, because as we know, a lot of the old-time breeders have passed. And so their children and things might not necessarily be into the plants as much, so then you kind of tend to lose those varieties, just the interest in those. So we've been going along. We did follow up with the suggestions that people had done last year on the survey to see if we could get some of the suggested varieties. But if anybody has any varieties that you think are important and would like to put in, please contact us, because we would really like to preserve these as much as possible. Yeah. Okay. And so I guess this is thank you to the Department of Agriculture for giving us this grant to enable this study. And it's been great to work with HFNA as well as Eric. So this is the workshop that he had talked about to kind of blend your scientific mind, your left brain and right brain. I'm telling you, it's stressful. It is stressful, because you're like, you're used to follow this protocol, this, but when it becomes the art where the spadix color should flow into your next thing, you're like, yeah. So it's stressful, but it's really good. It was a really good workshop, and we really appreciate that, because it's really nice to open up your mind and think of things. Seeing things from a different angle is always a good thing. And so I'm also going to give you an update too on how we also use part of these germplasms. And so basically, this is an old slide, but basically what we had developed, the industry had requested that we transform Anthuriums for bacterial resistance as well as nematodes. And so basically what we went through is we thought of it as a pipeline. And so if you can see here, we had the two varieties coming in. We go for bacterial resistance with Dr. Lisa Keith, nematode resistance with Dr. Roxana Myers, and then new novel traits with John Suzuki. And so basically we would have it flow out so we can get Anthurium plants for the industry. And before we used to joke that Dennis was here at this pressure point, and so he was the one that could add or release pressure. But so now what we're going to do is have Eric, in a nicer, sweeter way, provide that pressure for us in order to help the industry and move it along. So, okay, so just to back up, so we are talking about plant transformation, which is a biotechnology. And so what we need from this is we need to get the different genes for the desired traits in the plants. We need a plant regeneration system, because as Tessie, you had heard earlier from Tessie, some varieties are like awesome in culture, and some just kind of sit there. And you're like, come on. But, and then, so you need a regeneration system. You also need a method to deliver the genes into the plants. Okay. So, and then, so we're going to go back to your biology 101 class, just to give you what is a gene. And so, an important thing in the new transformations that we're doing is a lot to deal with this promoter region. And so, the promoter part of the gene, it kind of tells you where something will be expressed, how it's expressed, and when it's expressed, and how much is expressed. And your coding sequence is pretty much what trait are you putting into it. So you kind of have to find a balance of promoter as well as your important gene of interest. Okay, so we were working with some scientists from ARS, and so they had worked on monocot promoters. So as you all know, Anthurium is a monocot, which is more related to things like corn and more grasses, as opposed to a lot of the work that's done on some of your dicots, which is like your tobacco or bedding plants. There's a lot of tomatoes, a lot of different types of things. And so he had developed some different promoters that are from maize, from rice, and then the cauliflower mosaic virus promoter is one that comes from the cauliflower mosaic in vegetables. But this is more of a dicot promoter. And so most of the transformations previously done were with this cauliflower mosaic. Okay. So your promoter determines your expression of your genes, right? So as the cartoon says, there they go expressing themselves again, right? But basically what we're going to do is we insert this plasmid into an RNA and basically make a product. So because the transformation procedure takes a long time, and you saw how many promoters we had, rather than going for a more slower, stable transformation, what we're doing is basically blasting it into the cells and seeing which one expresses the most. And so basically what you have here is a gene ballistic gun. And basically what you have is you have pressure, gas coming down through the top. What happens is you have this rupture disk that when it reaches a certain pressure will release that amount of gas to go down. It'll hit your macro carrier, which you have your DNA coated on little gold particles. Okay? And then you also have a stopping screen. So just your gold particles with your DNA will go through, but the rest of it will keep it. And believe me, we know what happens when you forget the stopping screen is then you make a hole in your petri dish. So important to have the stopping screen. And then you have your plant material on the bottom. And so what you're doing is basically you're blasting your DNA that has your gene of interest into your cells and then leaving it for a couple days to say, what is the expression like? Okay, so now in this case, this is what we call a Gus reporter gene. And so basically it is just a gene that turns cells blue. So the more blue you see, the better the expression we're getting. And so here you can see the cauliflower mosaic. Right here is a blue dot, and right here is a blue dot. So pretty hard to see. But then if you compare that to some of the rice genes, or actually if you start looking at the rice ubiquitin genes or the maize ubiquitin genes, you see how intense that blue spot is. So that means we're getting better expression. And so let's say in the case of Anthuriums, where we're trying to do a protein that kills bacteria, you want it to be a little bit higher expression. Okay. And so here you can see the same thing, except in Anthurium roots. So you see the cauliflower mosaic virus here. So there is expression, but it's very, very light as compared to the maize ubiquitin and rice ubiquitin. So this is the one that Roxana likes, because roots and so for her nematode genes. So that's her favorite. Okay, so I told you that there's two basic methods of doing transformation. One is that bombardment, where we can do that transient assay that we showed you, and that is you just blast it in and see what the expression is a couple hours later. But then there's also another method that produces more stable plants. And this one, what we do is we use, I guess, nature's genetic engineer, and which is called agrobacterium. So what agrobacterium does is it normally has things in this TI plasmid called tDNA, where it can integrate it into the chromosomal DNA of the plant. And so this is what they do to tomato plants, where they produce a crown gall. So it tells them, hey, I'm going to put in all these genes that make food for me. And then the plant will produce the food, and then it'll eat it. And so that's kind of its mechanism of growing and surviving. And so basically what we're doing is we're replacing this part, this tDNA part, so instead of making that thing that makes a crown gall, we're putting in our gene of interest. Okay. And so these are different plasmids that we transformed into Anthuriums. And so these are the different antimicrobial peptides. Okay, and so for simplicity, we just call them D2A21 or D4E1. And so basically, remember I had showed you the transient expression where the blue spots was only where the DNA went into the cell. So now we're talking about more of a stable transformation. So the DNA has to go into the chromosome and be integrated into the chromosome. And so everything that grows from those cells will become transformed. And so here you can see how dark blue so this is a tissue that's been stained with GUS. And so you can see how dark blue it is, which means that our promoter is really good. Here you can see it on stems as well as the roots. So which means that using these promoters, we are getting we do have the genes being expressed. And so the antimicrobial peptides that we're using, were transformed into potatoes. And here you can see the wild type potato that's developing chlorotic symptoms. And here you see the transformed one. So it seems to work against the pathogen. And recently we went to ASHS, and they're also using the similar peptides to control citrus greening. And so they're finding promising results with these antimicrobial peptides. So what we did is then we checked it by PCR, because PCR will detect if it's actually integrated into the chromosome or not. And so we do have putative lines expressing the gene. And so if you see a line here, that means that we were able to amplify that gene from the plant's chromosomal DNA. And the untransformed control doesn't have it. So you know that these plants have the gene of interest. Okay. And so what we did is, because we couldn't wait, and poor Donna was kind of like, but everyone got so excited, so we're like, okay, well, let's try and inoculate it in culture before we do it. So we inoculated these with the Xanthomonas. We basically wait about 6 weeks or so, and then basically you take a different leaf, not the leaf that you inoculated, but a different leaf, and then you grind it up, and then you put it on the media for bacteria. Okay, and so here you can see this is the top one is the untransformed Marin-Seifert control. So what does that mean? So you see that the bacteria is this bright yellow and glossy thing. And so what does that mean is that means the bacteria went through the plant, is infecting the whole plant, and then now you're getting all this bacteria growing from there. So it's a high level. As opposed to, if you look at some of our transformants, so these all represent different lines, there's no bacterial growth, suggesting that it's actually killing the bacteria. So these are the lines that we're very excited about. And so these are the ones that we're multiplying and we'll be testing further. So yeah, and so they found similar results in citrus as well. So where the amount of titer or the amount of bacteria that you find in the transformed lines are much lower. So I just want to thank Donna, because she does all the work, basically, and our collaborators. So this is a truly collaborative effort, and so, yeah, just want to thank everybody. Okay, any questions? Yes?
That's like the amount of lines. What does that mean?
So basically, every time you do a transformation, you're hoping that it goes somewhere different in the plant's chromosome. And so that's what we call a line. And so the more lines, the better, because depending on where you integrate it into the chromosome, you could get less or more expression, or you don't want it to like jump into a gene that's for flower color or something, and then you'll be like, ah. So So that's kind of it. Yes.
What's the basic, John Q. Citizen difference between genetically modified organisms and CRISPR?
So CRISPR basically is you're doing a similar thing, but you're using genes from that plant, like internal genes, and basically you're turning off expression of a gene. So yeah, you're pretty much just editing it. So you're taking it out. So it's good for loss of function type of things. Like if you didn't want a plant to produce a certain type of toxin or a certain type of, or I guess in some cases you could use it for color. You don't want it to use a certain type of color or something, then you can just knock it out. Yeah, so those, because you're using only native genes, I guess they're not even considered being regulated anymore. Yeah. Mm-hmm.
So you're saying it doesn't have to be labeled as genetically modified?
The CRISPR, things that are developed through CRISPR. I think they're still going through a little bit of regulations, but as far as USDA, BRS is concerned, they don't see it as being an official GMO. Yeah.
CRISPR method, is that readily available to, or is that how they transfer knowledge?
It is. So the technology itself is, you are able to transfer it. Plants are a little bit harder because, and then in this case, it makes it a little bit harder for Anthuriums because you do require a phase where you do a cross, I believe. Yeah.
So what's a cross?
I mean, your hybridization. Yeah.
When you transferring it with a manual cross?
I think, John, if you help me, correct me if I'm wrong, but basically you're transforming it into one, and then you have to make it into a homozygous state, right? So then you're going to actually be selfing it, at least, or crossing it to something else.
You have like two components, and so one of the components that has to segregate away, that would be transgenic part, I guess.
Yeah.
And then you have to segregate it away from the mutation. And so it's kind of multi-step. You have to break down something, and then get segregated mutation.
So it's really good in some of the herbaceous or quick.
On the transgenic varieties, where do we stand timeline-wise?
Okay, so I'm guessing Donna's going, oh my gosh. No. So I think we're in the phase that we're multiplying it. We should be able to take it to the greenhouse sometime, I would say in the next couple months. And then hopefully we'll redo the testing then at that stage. And then after that, if it looks promising, then I guess we'll pass it on to Lisa. And then we'll get another a permit to actually do a field release for further testing.
To grow it out?
Yeah, that would be ideal.
So we gotta get ready.
Yeah.
Ready.
Good.
So what's it, about 3 or 4 years?
Hopefully a little sooner. Hopefully a little bit sooner.
I hope so too.
Yeah.
I want to be here.
Yeah. Yeah.
Long time.
Okay.
Okay.